Welcome again. My name is Evans Matuga. Today we are looking at cell physiology on osmosis. So I want us to understand what osmosis is in this particular lesson. And of course, as usual, I'll always be telling you that what we learn in class should be applied at your homes. You should be knowing how do you apply these concepts in your real life situations. So we are on osmosis. And before we define an osmosis concept, let's try to understand basically a few things about the cell. The cell, inside a cell, has a lot of things which includes organelles. And one of the organelles that is inside a cell is a vacuole. A cell vacuole or a subvacuole contains some salts and sugars. Those salts and sugars are what actually brings out osmoregulation in a cell. That is what you must have learned in Form 1 on a cell. So if a vacuole contains those ones, therefore any cell, any plant, any animal contains a cell. And any particular cell will always have a vacuole inside. So that vacuole will always contain salts and sugars. That is very important. Number two, this osmosis, aside from diffusion, this one, of course, it needs a medium. You know, diffusion occurs without using any root. It can just occur as amorphous. This one must have a medium. It must occur across a semi-permeable membrane. But before it again occurs, it is a process of concentrations. That we must deal with concentrations. When I'm speaking about concentrations, that should tell you that we can have higher concentrations and lower concentrations. What do they mean in osmosis? If I am so specific, I have been speaking of a vacuole containing salts and sugars. If I take a glass and this glass, inside this glass, I put salt and I put water and then I stir or swirl. What I'll have here, it will be tasting salty. In biology, if I bring another glass, and this glass now, I don't put any salt inside, it's pure water. Now this one will not be tasting salty, it's just water. So the one with salty, it is now called a concentrated solution. Concentrated solution. The one without salt is called a lowly concentrated solution. So now between the two, which one is higher and which one is low? So a salt one is having high concentration. With water, it's having a low concentration. The same case with the sugar. The one which has sugar is highly concentrated. The one with water is lowly concentrated. Now let me take you to your cooking of tea at your homes. When you have a tendency of putting more sugar and then it tastes more sweet, then that kind of tea is called a concentrated tea, a highly concentrated tea. A sugar without, tea without sugar, there's a name, we have it in my own language, I won't mention it. That kind of course, tea, it's now in biology called a lowly concentrated solution. Now you have understood. So it's not only sugar and of course salt that can make it concentrated. If you put more tea and it becomes more brown, that's more concentrated. The other one is less concentrated. So that is the same case. If you go to an hotel, you are taking some tea. Some hotels have a tendency of course, giving out tea which is having some milk, underline some, to customers. So that is a lowly concentrated solution. And some of them, of course, give a highly concentrated milk tea. And those are bigger hotels which you pay more. Those with 5 shillings a cup, of course, you expect to be given a lowly concentrated solution. Now, let's come back to now Osmosis. After understanding what is a lowly concentrated solution and what's a highly concentrated solution, those were very important for your own and better understanding. Now, let's define osmosis. Osmosis, therefore, is a process where water moves from their region of high concentration to their region of low concentration. Don't confuse yourself. A process by which water moves from their region of high concentration to their region of low concentration across a semi-permeable membrane, where water is high and where water is low. That should tell you something. Where water is high, that is more water. That is a dilute solution. Where water is low is less water. That's a concentrated solution. Do you get something? So therefore, osmosis is actually moving from where water is more to where water is less. That should tell you it's from a dilute solution to a less dilute solution. And that is why we call osmosis as a special type of diffusion, because of how water moves from their high region to their low region of concentration. Now, there are some terms that we must, of course, dissect here before we proceed. And they include the following. There are three. One of them is hypotonic. Number two is hypertonic. Number three is isotonic. Let's start from hypo. Hypo means low. So hypotonic is a lowly concentrated solution. The opposite of hypotonic is hypertonic. So hypertonic is a highly concentrated solution. And therefore now isotonic is a solution with same concentration. Same concentration. Isotonic. Using those two terms, hypotonic and hypertonic, we can now define osmosis. Osmosis can also be defined as a process by which molecules move from hypotonic to hypertonic region across a semi-permeable membrane. So from hypotonic to hypertonic. That makes better sense. Well, in a laboratory session, we can also test this process of osmosis using some experiments that we can have. And these experiments range from a number of them. And I will start from one. The first one that we can have is this one, using a whisking tubing. So that is why I've started with this one. Remember, when I was teaching you about diffusion, I used a whisking tubing. So don't you ever confuse yourself. You can be given a diagram like this and then you're asked, which physiological process is that? And then you're like, diffusion, osmosis. How do I answer this? Osmosis deals with concentrations. You must get that. So here, when you see a whisking tubing like this, and this content here should either be, this one can be, as of course, labeled here. Inside here we can have sugar, and then outside gets the water. It's distilled water. Then this is a whisking tubing, and that's a beaker. So what is going to happen if this is water outside and inside the beaker here, and inside the whisking tubing we have sugar? Remember, I told you that a cell membrane has got pores. It is semi-permeable. It is semi-permeable. It allows selective movement of substances in and out. So therefore, there is one which is going to go to some point. But here, because we are dealing with concentrations, we're not dealing with diffusion, we are dealing with osmosis. And we are dealing with concentrations. We are going to look inside the whisking tubing. Look at what is inside here. It is sugar. I already told you that sugar is a highly concentrated solution. Or if it is salt, it's also a highly concentrated solution. Outside here, where we have water, water is a lowly concentrated solution. So where do you think osmosis will take place? From where to where? Water, if it is lowly concentrated, we call it hypotonic. Sugar, we call it hypertonic. So therefore, the molecules of water will move from the beaker to the whisking tubing. And what do you think will be the result at the end of the experiment? There is increased content inside the fish king tubing. So that means this one increases. So it will now increase and rise. So there is a rise of content inside the fish king tubing. That is an observation as a student you are supposed to make in the laboratory when you are doing such an experiment. So this one can either be set as a diagram or you can be given as an experiment in paper 3 to carry out in your KCSE. This content is very important. And once you have understood that, it's now my pleasure to give you a few terms that you need to know before we proceed to the next. As we proceed to the next episode, it's now important for you to understand that there's another experiment which I'm going to give you, but it needs some terms for you to understand. There's a term known as turgid, oblique turgidity. There's a term known as flaccid, oblique flaccidity, or flabby. How do they come about? So there are two terms we are going to learn on plants especially. So a plant cell, if you put it inside water, it can either lose or gain. When a plant cell so loses that particular solution or water, it will behave in a manner that it is loose or soft, and therefore it will shrink. And therefore, in biology, we call that as a flabby. So such a kind of cell, we say it's undergoing plasmolysis. So it's plasmolized. But if it gains, we call that as deplasmolysis. So those are very important terms. So therefore, when we are going to look at turgidity, flaccidity, and all those ones, at least you have a clue, but I will explain more in the next episode. See you next.